Okay, good morning. Good morning, everyone. Welcome to this third session. Today with us we have Lara Nonell. Lara is head of the Bioinformatics Unit in Vall Institute of Oncology. She has a particular path, let's say. So she started mathematics, then did a master in information technology management, and got more recently a PhD in biomedicine. So what they do and what she's going to tell us today in her group, they analyze mainly genomic data and integrate different types of data. And the ultimate goal is to find biomarkers for cancer. So thank you, Lara, for being with us today. And
thank you, Arnau. Can you hear me properly? So thanks, Arnau, for this kind introduction. So as he said, I'm Lara Nonell, and I'm the head of the Bioinformatics Unit at the Vall Institute of Oncology. And today I come here to talk about transcriptomics. So because many of you might not have any idea what this is, I have prepared a very short Kahoot, okay? So let me, I know that you like Kahoots a lot, and this is just to break the ice, and this is a message for you that you can interact me at any time, and you can ask me whatever you, so please, I don't think the QR is going to work, but you can try. So it's just 4 questions on the topics we are going to discuss today. Okay. So you, so please tell me if you have any difficulties here. Okay, I think we are almost there. I don't know how many people are in the room, but might be. Any difficulties there? Shall we start? No. Okay. 35? Are we there? Okay. $38? Okay. Now, okay. Ready? Good. Okay. So you know, most of you know what transcriptomics is. Yeah, so it's genes. We will see that concept in a minute. Okay, well done, Eloy, Rick, Jan, Jana, and Carmen. Okay, let's continue with the. This is very easy. No option of no sabe, no contesta. Okay. There is still someone that doesn't know that microarray is a technology to assess transcriptomics. That's why I'm here. So Eloy, congratulations. Next one. Again. RNA sequencing. Is it a technology to assess transcriptomics? I think you have too much time to answer those questions. Wow. Yeah, this is another technology that we will see today. So, okay. Is Eloy a first-year student? Yes, okay, congrats. Okay. So should we care about lab experiments and all the things that happen in the laboratory when we get the data? Yes, of course. So this is the kind of, well, sorry, let me see who's. Okay, Rick, congratulations. Jan, congratulations. And then, of course, Eloy. Okay, congratulations to all of you. So this is, in fact, what we're going to talk about. So the first message is that there are several technologies that we will cover today, but those data that come from those technology have undergone a process in the lab, and all this processing have an impact on the data, and this is what we need to take into account. So the title of the talk today is From Microarrays to Spatial Transcriptomics, and this is, of course, from the oldest technology to the newest technology that study transcriptomics. So let me just see if this works. No? Oh, okay, it's a little bit, okay, sorry, I need to be a little bit more patient. So this is the outline. We will review what transcriptomics is and some of the technologies. And we will, at the end of the talk, we will see some of the best practices in analyzing this kind of data, especially on dealing with this kind of data. No, sorry, the other way around. So let's start with transcriptomics. And this is in the omics context, in where we study genomics, transcriptomics, proteomics, all of the omics that study the biology underneath, and that will bring us data that we as bioinformaticians will study, analyze, and take some conclusions. So as you know, and stated by the central dogma of molecular biology, the genes, so the DNA flows into the final product of protein in two steps, which is the transcription and the translation. So we will see that the transcription will bring us the product of transcripts, and this is what, of course, transcriptomics is studying. So we will start, as you know, I'm working in an oncology institute, and we study a lot the different cancers. One of the most studied and prevalent one is the breast cancer. So what do we have in breast cancer? So if we start from the end, so what we see is that we have many patients that have this type of cancer, and we're interested in understanding the clinical outcome and in personalizing the treatment for them, for those patients to have a specific treatment that will cure their specific cancer, because not all of the breast cancers are the same, and this happens with all the other types of tumors, and therefore we need a specific therapies and specific treatment for those patients. So to do that, we need this battery of different omics, and this includes imaging data, genomic data, transcriptomic data. Genomic data, in genomic data we are studying the DNA. In transcriptomic data we are studying the DNA after transcription, and that is the transcriptome. Then we have the proteomics data, and we have also the metabolomics data. We can do we can study different events in this omics data. We won't go in detail because we will focus on transcriptomics, but this is all the type of data that we can have. And this is the type of data that you can analyze at the end of the day. So, well, this is a lot of information. But where does this data come from? This comes from different samples. And when we are dealing with breast cancer, for instance, we can obtain samples from the breast from the tumor of the patient. And this is, we can study tumoral cells. We can study blocks of tumor that were preserved in paraffin. We can also analyze blood samples. And there is something that are called the xenografts. And the xenografts are mouse models. So it's mice where a piece of tumor was inoculated, and this is very easy to test different treatments. So we use this kind of modeling, the xenografts in mouse, because this allows us to preserving the ethics of the animals, which are different from the ethics of the human. We can test different treatments, and this is very convenient. So there are several scenarios in where, sorry, I'm screaming too much. So there are several scenarios in where you have different mice. In each of them, you have a specific a piece of tumor, so you can try a specific treatment, and you can try which is the best treatment for this specific patient. So this is very convenient, and this is of course something that you cannot do in human, and that's why we do that in mice. So as you can see, there is a myriad of different types of samples that we can analyze using different omics that will lead to different omic data, and this is what will arrive to you. So you need to analyze, you need to understand the biology, a little bit the biology underneath. So, sorry. So let's focus in transcriptomics, but first of all, we need to know what a gene is. So just a gene is the basic unit of hereditary in living organisms, and is a portion of DNA that contains coding and non-coding sequences, and only the coding sequences will be there after transcription. So this is something that you can see. This is quite small for you probably, but from the coding sequences that are called exons, they will be transcribed, and this is what we are going to study, so what we call the transcripts. And how are those transcripts transcribed? So under specific conditions, cells are presenting different types, so they are expressing different genes, and those are the ones that are being transcribed. So by analyzing the genes that are being transcribed, we can understand the mechanisms, and we can also identify biomarkers to define targeting therapies, for instance. And this is something that we need to understand. So from gene, we are going to be interested in the transcripts. Those are the ones that are being expressed in a specific cellular process or under specific conditions, which might be, for instance, the cancer condition. So I know. Okay, so transcriptomics is going to be the study of all those transcripts, and for that we need specific high throughput technologies. For instance, microarrays. There are several other types of technologies, such as quantitative PCR, sequencing technologies, and even spatial transcriptomics that we will see today. So which are the applications that we have for transcriptomics? So we can have, of course, we can study gene expression under specific conditions, and that means that we will use usually comparison studies. We will compare different conditions. If we focus on cancer, we will compare, for instance, tumoral samples against normal samples. We can compare tumoral samples treated against tumoral cancer non-treated. And this is what we are going to do in general in transcriptomics. We are going to compare, and that's why we need to define properly the different conditions that we are going to compare. So we can, we can also perform alternative splicing, and that means that we can study the composition of the different exons that are being transcribed. We can perform functional enrichment, and this is beyond the expression of a specific gene. which are the functions that are doing these transcripts that are being expressed. So are those cells migrating? Are those cells dying? And this is a process called apoptosis. What are those cells doing? So we can either study the genes, so the transcripts, or we can study the different functions. Out of this, we can, of course, discover biomarkers that might be involved in biological process, such as the cancer condition, or gene signatures that are that is a set of genes that can be involved in a biological process. We can perform classification of samples, and this is something that we will see related to breast cancer specifically. We can generate predictive models. We can study pharmacogenomics and toxicogenomics, and that is how is treatment, so the dose of a treatment that we need to administer to a mouse, to a person, to a patient, to have the best results. So back to our main slide of breast cancer, I was talking a little bit about the different samples. I've commented that we can obtain samples from blood. We can obtain samples of the tumor itself. We can use this tumor to inoculate a piece of it in a mouse and try different treatments. So all these samples are important because those samples will define later on how those samples are processed in the laboratory for a better performance, for the best performance, let's say. So the message here is that the nature of these samples will condition the processing that we can conduct on those samples, and ultimately in the bioinformatics data that we will analyze. So let's start with the technologies, and for that I'm based on a figure that was published by Hong et al. A couple of years ago, in where the different technologies was placed in a timeline, and starting with the double helix, which was discovered, let's say, by Watson and Crick in 1953. Then we have the Sanger sequencing, which was the first technology to obtain the nucleotides of the different DNA. And in this Sanger sequencing was applied to to sequence the human genome. In the meanwhile, the microarray technology appeared, and this was in 1995. And a couple of years after the human genome was sequenced, the first technology for sequencing appeared, which was the peer sequencing by Roche, and this was in 2005. And then a couple of other technologies, a couple of other sequencing technologies appeared. And Illumina, which is now the gold standard of the sequencing, appeared at that time. It was a company called Solexa, which was bought by Illumina, and they created the first sequencers, which is the MiSeq and the HiSeq, and those are still in use in many, many genomics departments. So in 2009, the single cell appeared, and that is the study of the transcriptomics of just a single cell. And then lately, we have the spatial transcriptomics that we will see that can allow us to study the transcriptome on a spatial slide. In addition, we have the Oxford nanopore that appeared in 2015 and allow us to sequence long reads, that we will see in a minute what that means. So in general, and this is important for you, because here we have bioinformatics, and the whole process starts by creating a proper experimental design. That means that the investigators, and luckily you will be someday involved in this process of the experimental design, because this will condition how you receive the data. So if you can be involved at the beginning of the process and really counsel on how the conditions which are the conditions that you need to study, how many samples you need to have in each group. So here I have placed, for instance, 5 tumoral patients, and so 5 cancer patients and 5 normal patients, or normal people, sorry. And this is what we want to compare. We want to obtain the biomarkers that distinguish the breast cancer from normal population. So for that, we need to create a experimental design. We need to extract the RNA, and for that, there are specific protocols. We need to control the quality of these samples, and this again will condition the type of the protocols that we will apply in the laboratory later on to process those samples, and this is the sample processing. Depending on the amount, on the quality, and on the type of data that you have, or the sample that you have, this will condition the processing of these samples, and this will condition later on the bioinformatics. So all those steps that are here will condition the data that you will analyze later on. So imagine all the things that can happen in these steps to get bad or good data. So, and finally, we need to validate all these results after you have performed the bioinformatics analysis. So back to our technologies. So let's focus on microarrays. Microarray is just a collection of biomolecules that are called probes and are arranged in a matrix design on a solid support. The nature of those probes will define the type of array. In this case, we will focus on cDNA microarrays that will study transcription. Okay? So this is a microarray. This is a little box like that. And this contains one transcriptome of one patient, of one person. Can be a patient or not. Okay? And the probes need to be pre-designed in each of the different spots that are here. So this is like a matrix with different spots. Each of these spots has pre-designed probes, and this is targeting a specific area in the transcriptome, okay, in a specific gene. So by studying all those areas, we can study a complete genome just by targeting specific areas. So this, if we have, this is a genomic locus, and those are the different transcripts. So we can here, those are the different hyphens that you see here, these different lines that you see here. Those are the probes that were pre-designed to interrogate these different transcripts. There are different technologies inside the microarray world that will allow us to focus on some exons or in some specific transcripts. So once the array is designed, the samples undergo a specific processing that implies fragmentation, amplification, and many other steps in the lab. And then those microarrays are finally scanned here, and we obtain those images. Those images contain different spots, and the intensity of the spot is the amount of transcription that we have for this specific region in the transcriptome. Okay, so the highest the intensity, the more transcription we have in this area. So the more expression we have for this gene, and what we have is different scans that we ultimately transform into a continuous matrix. So what you will analyze at the end of the day is a matrix that contains genes in rows and the different samples in columns. And what we have is the measurement of all those genes for all the samples that we are interested in. And we need to define different methods, and this is something that we have here. We need to define bioinformatics to analyze these matrices. How do we start with these matrices? So we start by usually doing a quality control on the data. We have performed quality control on the samples. We have performed quality control on the arrays. We have performed many quality controls. But now we have those matrices, and and we need to specify or to perform a quality control. Then we perform preprocessing, and that means that we get rid of those probes that did not work properly or that are not interesting for us for the specific process that we are going to follow. We need to normalize to take all those intensity to similar levels for the different arrays to be compared. And then we perform differential expression analysis. And this is what I talked about, the comparison analysis. In transcriptomics, we always compare different conditions. And the goal is to perform differential expression analysis. That means that we are going to compare the microarrays of the tumoral samples that we had here in the okay, so we are going to compare the microarrays of these 5 cancer tumors and 5 normal samples. And we are going to we are interested in the profile of those cancer types, those cancer samples, because we want to know what's going on in this biology, what's happening in these cancer samples. So this is the differential expression analysis that is performed here. One of the plots that are used mainly is a heat map that is showing us the difference of intensity. Do you know this plot? The heat map? Anyone? So what it's showing is here the differences of the conditions. So we have 3 here samples that might be tumoral and non-tumoral, and those are the profiles of, in rows we have the different genes. And here we see the intensity. In red is high intensity, in blue is low intensity. So these samples here have a block of genes that are low expressed, and those are high expressed, and the other way around for this normal sample here. We have also the volcano plots that is accounting. Here we have the more differentiated expressed genes between the conditions that we are studying, and here taking into account statistical parameters. And finally, after the differential expression analysis, what we usually do is to perform functional analysis. And that is what I was commenting at the beginning, is beyond the gene analysis is a function analysis. So we are interested in what is the enrichment? What are those genes doing in the biology? Are they migrating? Are they the cells dying? What is happening? So this is what we do in the functional enrichment, and we have a specific bioinformatic tools to do that. We work basically with R and Python. So just an example of how microarrays can be applied in the cancer field. There are thousands of different articles, and we will see later on how this technique has evolved along the years. And this is a very well-known classifier, which is the PAM50 classifier for breast cancer. And this was originally published in 2000, and they detected out of the breast cancer samples 4 types of cancer. One was, so the original publication was by Peru et al. In Nature in 2000, but what they defined is the same group. The first author was Threes Sorley, but it's the same group. They managed to identify by using heat maps, the plot that I showed you before. They identify different types of breast cancer. And this is the basal-like, which is the one that has the worst overall survival. And we have the luminal A, which is the one that is having the best overall survival and relapse-free survival, of course. And then we have also HER2 positive and luminal B samples. So they managed to distinguish by using microarrays different types different types of breast cancer, and this is very much used in the clinic nowadays. So now we use other technologies to obtain these different types of breast cancer, but still this is the origin of the PAM50 classifier. So there are mainly in breast cancer two types of classification. One is the molecular subtypes, which is the PAM50 that I just presented, and then we have the clinical subtypes, which are the hormone sensitive, HER2 positive, and triple negative breast cancer. A lot of research has been done on the classification of the different types of breast cancer led by these molecular subtypes or intrinsic subtypes, but now, because they have a bad response to triple negative breast cancer patients, there is a lot of research done on this specific group. So this PAM50 classifier, this molecular classification, is used nowadays in commercial application. That means if you know anyone that has gone through a breast cancer, probably one of those assays has been performed on the tumoral samples, and this is Prosigna that measures the risk of relapse, and this is based again on the PAM50. And then we have two other classifiers, which is the Oncotype DX and the MammaPrint, which is especially a design based on microarrays, not on the PAM50, but also on microarrays, using the same methodology to study the prognosis and the treatment for breast cancer. So a lot of this research that we perform in microarrays and in the other transcriptomics technologies will have an impact on the clinic. So let's focus now on the sequencing technology, RNA sequencing, that some of you didn't know what it is. So the RNA-seq is the combination of high-throughput sequencing with computational methods, and this is where you appear, just to capture and quantify the transcripts present in an RNA sample. This can be done in two different, basically using two different technologies. Short reads, which is the old technology that has the reads, what they call reads of usually between 100 and 200 base pairs. And this means that we are going to read each nucleotide of this sequence, and we are going to know in the genome where it's placed. And then we have long reads. Long reads are more than 1,000 base pairs, and this is very convenient when you don't have a special bioinformatic pipelines or a lot of different equipment in the lab. So we can also distinguish different technologies for short reads and also for long reads. But basically what we do is we amplify the sample. Well, in general, there are many technologies here, but we fragmentate and amplify the sample, and we obtain different fragments. And those fragments undergo to a flow cell process in the Illumina technology that will tell us whether this is an A, a T, a C, or a G. And this is what you know that belongs to the DNA. So once we know the different technology, once we know the different sequences, we can infer these into the genome. And how do we do that? So following the same pattern that we use for the microarrays, in here we have the different exons of a gene, the different transcripts, and what we have in short reads are these reads. This is ACTG chains or sequences that we align to a specific area of the genome. And that's when we can know the amount of RNA that is being transcribed here. So the more sequences I have in a specific area, the more transcription that I have, okay? More the cell is producing more transcripts on this specific gene. For long reads, as we have the sequence is much longer, we can really identify usually just a piece of a gene directly. So at the end of the day, what we do is, specifically for short reads, which is the most broadened technology in RNA-seq, what we have is a counts table. And that means that we have, again, a matrix of genes and samples. But instead of having the intensity in the matrix, what we have is the read counts. So for each gene, we will know how many, for a specific sample, we will know how many reads we have for a specific gene. So the processing is very similar to what we did for microarrays. So in here, the only steps that we are adding is the alignment and the expression quantification. So we need to align those sequences to a reference genome, and this is something that you saw in the previous talk by Tyler, as far as I remember, and this is something that we do with these little fragments. We align, we quantify, and that is when we create the expression matrix, the counts matrix that we saw previously. And then we perform differential expression and functional enrichment the same way as we did for microarrays. Okay? Of course, the statistical methods that we apply in this context are different from those that we use for microarrays, because in microarrays we had the continuous intensities, and here we have counts. Okay, so statistics is different, but the objective is the same. So some of the applications in this field are, of course, the expression that we have seen. We can also analyze alternative splicing. We can study fusion genes, that is, genes that instead of being complete are getting connected to other genes that are in other places of the genome. We can also study the mutational profiling, all those errors that might occur in the different reads. And we can also deconvolute the cell types. That means that we can know whether we have lymphocytes, we have granulocytes. This type of information can be deconvoluted from blood samples, for instance. And here, this is an example of the Oncotype DX, Mammoth Print, and PAM50 performed on RNA-seq data. So those classifiers that were done in using microarrays can be also done in RNA-seq. Okay? So, and this is very convenient also in the clinical practice. So what is the advantage that offers RNA-seq on the microarrays? So for microarrays, we needed to define a genomic region to define the probes. And those were going to the microarray, and those that were the predefined regions. We don't need to do that anymore because we are going to sequence everything. Okay, so we will have all the information for all the data. We'll have low background, and we will have more information that we as bioinformaticians can use more in detail. So let me focus now on single-cell RNA-seq. What is the single-cell RNA-seq? So this is just a combination of the RNA-seq into just a single-cell level. So we are going to extract a single cell, just one cell, and we're going to study the transcriptomics of this specific cell. There are several preparations, there are several methods to extract these cells, and it is a little bit tricky because most of the technologies that have been developed to extract those cells have different limitations. One is then when you extract one cell, because cells have different measures, it can be that instead of studying one cell, you study two cells. Or it can be that when you are using those doublet, or there are many technologies that you don't have anything there. It is a little bit tricky to make sure that you are studying just one cell. But if you have that, you have one cell, and then you can study the transcriptome of one cell. this is the different processing that is to obtain a single cell, and then we perform the sequencing that we have seen before. So what is the processing here? So we obtain also count matrices, but this is going to be one matrix for each single cell. And what we are interested here is in clustering those cells to see which are similar to which ones are similar, and to identify different types of cells that might be doing a specific process. We can also perform, there are many other things that we can do, but once we have done this at the single cell level, we can compare between different cells, and we can identify different clusters and perform, as usually, the differential expression and functional analysis. And finally, well, we can characterize, using single cell, we can characterize subpopulations in a specific block of tissue. We can perform cell differentiation and this is an example, again, of breast cancer that you can study and ask me if you have doubts. And of course, this has an advantage over the RNA-seq traditional, that we are going to study just one cell, and we are not going to analyze the whole bulk RNA. So just a couple of words on spatial transcriptomics. This is just a combination of imaging and RNA-seq. And this is, we have different slides of a tumor, and we can analyze the different cells at a spatial level. So we can take different cells there and analyze the transcriptome of those cells on a spatial basis. So here we are going to combine bioinformatic methods for imaging and for sequencing. And this is again an example. So let me recap here. So we have the first technology, that is the microarrays technology, that was analyzing just different probes. Then we have the RNA sequencing that can allow us to analyze different areas, so the complete transcripts. Then we can do that at the single cell level, and then we can also do that for spatial transcriptomics. So all those parameters will condition the type of technology that we are going to use. And especially, this might not be you who is going to choose this technology, so you need to be aware of those differences among the technologies. So this is just a curve on the use of those technologies in PubMed. So nowadays, what is really being in use is the RNA-seq, and the single-cell RNA-seq are gaining the floor there, and microarrays is something that is going down. So just a couple of words on best practices. We need to be aware that when we are performing bioinformatics, what we do needs to be reproducible. And for that, we need to be aware of different standards and follow ethical principles. For that, there was a project, the MacQC, and this involved the reproducibility on microarrays and also on sequencing when we are analyzing the data. And this is crucial because if we give one data set to one bioinformatics group or to another, you will get different results. And this is something that we need to be aware. And this is something that the MacQC study addresses. And they show the differences of giving data to different bioinformatic groups and obtaining different results. So it's quite interesting. This is about the data and about how to publish data. You have it here. And I just want to give you a brief overview of what we do, okay? So this is what we, in practice, do by the Vall Institute of Oncology. So we have a cluster here. We analyze data using Docker and Slurm. We use Singularity as well, and NF-Core pipelines. Those are the bioinformatic pipelines that we use to process all the types of omics data and to integrate this omics data. We also use the cloud. We have our code in GitHub in bioinformatics, and we use standards for programming. We try to follow them. We do our best to follow them, and we program basically in R and Python, and we use some tools for project management. And we follow those standards, and we upload the data. When we have analyzed the data, we need to upload this data to be published. And yes, this is our service, and this is what we do. And so this is, so what we have been focusing. So we do some stuff, and sometimes we as bioinformaticians, we don't care about the other stuff, which is also very important. So the message here is everything that is done in the lab, in the transcriptomics and other omics fields will condition the data that you will finally analyze. So that's it. If you have any question.
Thank you, Lara. Thank you very much for this fantastic talk. So time for questions. As always, we will start with people which has priority for today's session. Later, we will open to others.
How do you know that mouse recreates correctly the conditions of cancer in humans?
So we don't really know, but all the models have been created in mouse. And this is because of, well, there is a huge orthology between the two species. And we are based on that orthology to test the different treatments in mouse. But there are other model species that are called, like zebrafish and many other model species.
Okay, so you focused a lot in breast cancer. So right now or in the near future, can these technologies be applied for all types of cancer? If not, what factors are involved?
Yeah, it is applied to all types of cancer, but I didn't want to show you, I wanted to focus on just a specific cancer, not to lose the field. But all this technology is applied in all types of cancer. Yeah, bladder cancer. There is a very huge project, which is the Cancer Genome Atlas, and this is an atlas of different types of cancers. And they use transcriptomics, genomics, and methylomics, proteomics. I think.
So you have talked about microarrays, but I haven't understood at all how microarrays are applied to the transcripts.
So in fact, what we have is in the microarray, let me put the slides. Why are you hiding? Did you ask? Who? Ah, okay. So, no, I think, let me put the slide of the microarray. So what is not clear is what is in the microarray, probably. Yes. Among others. Okay. Now, okay. So here we have artificial probes that interrogate a specific place in a genome. So like gene ABC, so just a specific area of gene ABC. When we prepare the sample of the breast cancer, we have a preparation of the transcript there. We have fragmentation, we have done the amplification, and we have a loads of amounts of transcripts there. When we have this species of the breast cancer sample, we are going to hybridize that into these predefined regions. Those that match, that hybridize to those that are inside the microarray, will give us the amount of transcript. So the highest number of those material coming from the breast cancer that is hybridizing in this microarray will give us the highest intensity. It is a little bit more clear. So this image here, each spot is the amount of transcript that you have for this gene. Okay, so gene ABC, this spot here. Yes? Okay.
So, at the beginning, Arnaud mentioned that you started studying maths.
Yeah.
And my question was, what were the reasons to pursue such a different career path after finishing your studies?
Well, my career is a little bit atypical. I studied maths. I studied computing science later on, but I was working for consultancy companies. I moved to Germany, and when I came back, I realized that I didn't like the consultancy companies, that I wanted to do science, and so I started my PhD. I had two children, and yeah, that's, and at that time, no one was talking about bioinformatics. We were talking about statistics, data analysis, data mining, because I'm rather old. So, yeah, I started my PhD by combining statistics and data mining, and that's why I arrived here.
So I want to specify in one question that you more or less has been asked. So in microarrays, you said that it has been identified different types of breast cancer, but it has also been identified other types of cancer, like lung cancer or colon cancer, like other types of that.
Yes. Yes, it's a similar answer. So I focused on breast cancer because I didn't want to use different examples of other types of cancer, and because breast cancer is one of the most studied one. It's very much prevalent, but yes. So I'm going to show you, if I manage, can you do the next question, and I'm going to just show you ups.
Hi. I wanted to know, these technologies, aside from cancer, which one would be the best to study different types of illnesses, and why?
Well, this is it depends on the objective that you have. But as you have seen, RNA-seq offers us a lot of advantages because you have a profile of the transcriptome in just very quick, and the technology is very much spread. So you can find different genomic facilities everywhere. But if you're interested, but for RNA-seq, you are just studying bulk RNA. That means that you have a sample, blood or tissue or whatever, and you extract the RNA. You don't care about the cells, but cells are very different. And if you're interested in a specific type of cell, then you would need to go to single-cell RNA-seq or to spatial transcriptomics.
And then in which case would you use the single cell? Like an example of that.
Like an example. So when we have, we have, for instance, some investigators that have, are treating the cells, and they have found that some of the cells remain senescent. Senescent are those cells that remain not dead, but they are calm, and all of a sudden they develop a new cancer. So we use single cell to identify those senescent cells because those have a specific expression for specific genes. They have genes that have high expression. So we need to detect those at the single cell level.
I just wanted to ask, how do biomarkers indicate the type of treatment that suits each patient?
So what you mean how to develop? So in general, we identify the processing in research is you identify biomarkers, and those are maybe going to be developed as a treatment. So am I answering your question, or?
Yeah, I just wanted to ask if a certain type of biomarker was present, then a certain type of treatment was given to the patient, or?
Yeah, but this is not called biomarker. So no, well, yeah. So yes, that's a very interesting question. So in some cancers, the presence or the high expression of this happens in breast cancer when you have the hormone receptors, and those are specific genes, the progesterone receptor, the estrogen receptor. When those are present, you are given a specific therapy. When you are not having those genes expressed, then you are not giving these therapies, you are giving others. And this is conditioned by this classification that we saw.
Thank you.
You're welcome.
Thank you very much, Lara, for your talk and for the answers to the questions. So yeah, let's thank again.
Just one thing that I was looking for. Those are this is the Cancer Genome Atlas, and 33 types of cancers were analyzed using different technologies such as transcriptomics. Okay, so not only breast cancer, all types of cancer. Sorry. So good luck in your career. If
you know, statistics is important, so please rush to the Hafid's class.